Good morning everyone. I am Dr. Kalpana Sharma, Department of Zoology, S.P.C Government College, Ajmer. बच्चों, मेरे लास्ट लेक्चर में मैं आप लोगों को बेसिक कंसेप्ट्स ऑफ एनिमल सेल एंड टिश्यू कल्चर के बारे में बता रही थी, जो कि आपका बीएससी पार्ट सेकंड का थर्ड पेपर है। उसमें मैंने आप लोगों को क्या क्या चीज़ें बता चुकी थी कि टिश्यू कल्चर क्या होता है, प्योर कल्चर क्या होता है, वो कौन कौन सी रिक्वायरमेंट्स हैं जो कि कल्चर टिश्यू कल्चर या एनिमल सेल कल्चर के लिए असेंशियल है, लेबोरेटरी रिक्वायरमेंट फॉर इन विट्रो कल्चर्स क्या क्या होनी चाहिए, मीडियाज कौन कौन से अवेलेबल होते हैं। आज के लेक्चर में मैं आप लोगों को प्योर कल्चर मेथड्स के बारे में बताने जा रही हूँ। प्योर कल्चर मेथड टू ऑब्टेन अ प्योर कल्चर ऑफ एनी माइक्रो ऑर्गेनिजम्स a single cell must be introduced into a sterile growth medium. सिंगल सेल को हम स्टेराइल ग्रोथ मीडियम के अंदर डाल देते हैं अगर हमें किसी तरह का भी माइक्रो ऑर्गेनिज्म का प्योर कल्चर करना है तो। बाय डाइल्यूटिंग अ सैंपल, सिंगल सेल कैन बी ऑब्टेन्ड दैट कुड ग्रो टू प्रोड्यूस अ प्योर कल्चर. 3 different methods are there to obtain pure culture, which are namely number 1 pour plate method, number 2 street plate method, number 3 spread plate method, and number 4 special methods. Microbiologists begin now finding the possibility of diluting on a solid surface. It is called as streak method. Streak method was pioneered by Robert Cook. In pour plate method, number 1, the diluted sample is mixed with the previously melted agar growth medium at a temperature just above the solidifying point. Number 2, the mixture is poured into a suitable vessel and is incubated. Number 3, each cell produces a colony within the agar media. If the organism is to be isolated, is present in the large numbers in the mixture, then serially diluting the fluid or sample with sterile water to the point of extinction in numbers of cells. For example, suppose 10 ml of a soil sample in water is there and it contains 1000 microorganisms. It means 100 microbes are present per ml. If we take out 1 ml of this and mix to another 9 ml of sterile water, there will be 100 microbial cells in 10 ml or 10 microbes each ml. That would contain a single microorganism. There are some special methods also. There are some special methods which are used only for restricted type of microbes. ये जो methods हैं, special methods, उनके नाम हैं enrichment cultures, single cell isolation, 3 selective media. First of all, we are going to see the enrichment culture. Enrichment culture क्या होता है? This method is used for those microbes which are small in number or small numbers in a sample, number 2, and whose growth slows than other species present. Culture may be selectively enriched by varying the composition of the medium and the condition of incubation table. As you can see from the table, this table is showing the enrichment conditions for Chemoheterotrophic bacteria. The components of the mediums are given in grams per liter. Incubation at 25 to 30 degrees centigrade without illumination. So you can see from this table, addition to basal medium, organic or inorganic, spatial environmental condition, atmosphere or pH, and organism enriched. Next is your single cell isolation. Single cell isolation can be isolated by a micro manipulator by which very fine glass pipettes can be manipulated mechanically under the microscope. Next one is the selective media. Such media contain specific chemicals which do not affect the growth of the bacterium to be isolated but discourage the growth of bacterium to be isolated but discourages the growth of other bacteria in the mixture. For example, addition of sodium azide into media selectively isolates the lactic acid bacterium. Blood sugar is the most commonly used differential medium. Certain organisms have the ability to lyse RBC. If RBCs are completely lysed, a clear area surrounds the colony. This is called as beta hemolysis. What is beta hemolysis? If the RBCs are completely lysed, a clear area surrounds the colony, which is known as beta hemolysis. Besides beta hemolysis, there is alpha hemolysis. What is alpha hemolysis of RBC? Partial destruction of RBC produces a green opaque zone that is termed as alpha hemolysis. Besides alpha hemolysis, there is gamma or non-hemolysis. What is gamma or non-hemolysis? Organisms that cannot destroy RBC are gamma or non-hemolytic bacteria. Now next we are going to see the media for different cultures. First is the enrichment media. This medium encourages the growth of a particular organism which would like to be overgrown by competitors in a simple with a complex mixture of microbes. The desired microbe becomes the dominant species because it is better adapted than its competitor. Second media is the enriched media. What is an enriched media? Media containing the nutrients required to support the growth of a wide variety of organisms. In addition, they contain nutritionally rich supplements such as blood, chocolate, serum, etc. Third is the transport media. What is a transport media? Any medium that is used for temporary storage of specimens while they are being transported to the laboratory for examination is termed as a transport medium. This medium ideally maintains the viability of all organisms in the specimen without altering their concentrations. Next media is the enumeration media. What is an enumeration media? These are the medias used for enumerating bacterial contents of such materials as milk and water. Next is the characterization medium. This medium is mainly used to determine or characterize the type of growth produced by the bacteria as well as to determine their ability to produce certain chemical changes. For example, gelatin stab. Last one is the selective media. What is a selective media? Such media contain specific chemicals which do not affect the growth of the bacterium to be isolated but discourages the growth of other bacteria in the mixture. For example, addition of azides. आज के लेक्चर में मैं आप लोगों को डिफरेंट टाइप्स ऑफ ये स्पेशल मीडियम्स के बारे में बताया. मेरी नेक्स्ट क्लास में मैं आप लोगों को एनिमल सेल कल्चर के बारे में बताऊंगी. थैंक यू.